Okay, so I am super excited for today's talk, which is going to be given by Sara Dell Williams. Sara she got her PhD in ecology, evolution, and marine biology from Northeastern University. And she uses network science and modeling combined with fieldwork and physiological experiments to attack problems facing coral reefs from multiple angles. She studies the connections among polyps in coral colony, the associations of coral species in a symbiotic algae and microbiome, and the spread of coral diseases, all under the lens of global climate change. Sara is currently a postdoc at Coral Health and Disease Lab at Mote Marine Laboratory in Sarasota, Florida. So Sara, take it away.
Okay, great. Thank you so much, Alice, and thanks for having me for this seminar series. So today I'm going to be talking about the resistance and resilience of coral-algal symbiosis networks. So first, I just want to make the case for coral reefs as complex systems that are suited for network analyses. Now, a pretty good definition of a complex system is an adaptive system with many interacting parts whose behavior cannot be predicted by understanding of subcomponents of the system. So a coral reef is extremely biodiverse, so they have a lot of interacting parts, and they're interacting in a lot of different ways, from predation to competition and symbiosis. And we can even think of human society as interacting with coral reefs when we consider all of the benefits we gain from them, like important fisheries and revenue from tourism. Now, these coral reefs are also adaptive systems because they are facing and responding to multiple threats, from increasing temperatures and acidifying oceans due to climate change, and to other direct anthropogenic stressors like overfishing and pollution, and also disease outbreaks. So they're adaptive, they're responding to these stressors. And then lastly, we can't predict the behavior of a coral reef or its outcome when it's adapting to these multiple stressors by just considering one species, one environmental condition, one location in the ocean. We have to take a complex systems approach to coral reefs to better understand either their persistence or decay while under all of these attacks. So we could also just ignore all of the other organisms on a reef and solely focus on the coral, and we would still have a complex system. Now, just to be clear, corals are animals. They're not a plant or a rock like many people tend to think at first, so they're animals. And then on a reef, there's many individuals of different species of coral. And this is the coral animal. It grows and lives on top of its own skeleton. So yeah, there is a rock part to the coral. And an individual coral is referred to as a single colony because it's made up of a network of individual units called the polyps. But that's a talk for another day. I won't get into that network. So corals also have a symbiosis, a beneficial relationship with algae that live inside their tissue. So these are like tiny little plant cells that live in their skin. So they do have a plant part. And this symbiotic algae gets a home and nutrients for photosynthesis, and the corals get an extra energy source, that photosynthetic product. And they also, the algae give coral their beautiful colors. Without the algae, they would look white like their underlying skeleton. Right, so on a reef, you have many different individuals, all these individual coral colonies that all associate with different species of symbiotic algae. And the corals can actually host more than one species of algae at a time. So we can analyze these diverse associations as bipartite networks of coral hosts and algal symbionts, and then put them into the context of their environment where they are constantly facing perturbations and stress. So a central goal of coral reef ecology is understanding how this complex symbiosis responds to changing environmental conditions. To kind of simplify this area, I like to set things up in this equation. So a network plus stress equals question mark. It's just my framework for understanding the system. So when we study how a system responds to stress, we are usually asking how resistant and resilient is a system to a perturbation. And that's what this equation is kind of setting up the framework for. So while resistance is commonly accepted to be the ability of a system to withstand or resist a disturbance, or we could even say it's the ability to tolerate a disturbance or stress until the loss of functionality occurs. The definition of resilience tends to vary a lot across disciplines. So I'm just going to define resilience as the ability to adjust system activity when faced with disturbances or stress and recover to a functional state of persistence. All right, and I explore the resistance and resilience of the coral-algal symbiosis through 2 different projects. So in this first one, I'm looking at the resistance and robustness of the global coral symbiont network. So just the resistance part. And really, I'm asking how resistant is this global coral symbiont network to perturbation and why? What is happening during the stress to the point of collapse of the network? And this work is published in Ecology. And in the second project, it builds upon the first by now also considering the potential for the coral symbiont network to recover from stress. So I'm asking what happens during and after stress on a local coral symbiosis network in Bocas del Toro, Panama. So is the reef resilient? So I break down resistance and resilience of the coral-algal symbiosis into these 2 different projects. Jumping right into the first project, remember on a reef, there's this diverse pattern of associations among corals and their algal symbionts. These algal symbionts are in the family Symbioninaceae, and we can consider the network of coral species and these symbiont species on a reef, where individual corals can associate with multiple types of algal symbionts, and different species associate with different symbionts as well. Using the GeoSymbio database, I mapped the global network of coral species in Symbioninaceae. Now, this network included 360 coral species from 14 subregions of the ocean. So if a coral species was found in different subregions, there's actually multiple nodes for that coral species. So say we found a staghorn coral in the Eastern Caribbean and also in the Western Caribbean, there's 2 different nodes for that coral species, one in each location, because those coral hosts are experiencing different environmental conditions. And I wanted to make sure that was incorporated into the larger network model. So then along with the coral species in the networks, there's 250 symbiotic algae types that represent 4 different genera of Symbiodiniaceae. There's also 1,697 links in this network. It does have a heterogeneous structure. The degree distribution follows a truncated power law. So there's many specialist species and a few generalists. And these generalist hubs are the symbionts. All of the top 3 hubs in the network are from the genus Cladocopium. And then most coral hosts only have 20 or fewer associated symbionts. So much less connections than their symbiont counterparts. So this network is under attack by increasing ocean temperatures. When it gets hot on a reef, corals experience coral bleaching. And coral bleaching is when the coral gets really stressed because it's too hot and no longer wants to deal with its symbiotic partner. It just says, get out of here, I can't handle you right now, it's just too hot. So they kick out their symbionts, and then they no longer have their beautiful colors and they look white like their underlying skeleton. So once they kick out those symbionts, they're no longer getting that extra nutrient energy source from photosynthesis, and they have to capture all of their food themselves and eat it that way. So they're kind of working themselves to death in this like really hot environment. So corals often die from coral bleaching. But there's also a lot of variation in coral bleaching. So different species bleach at different rates. Maybe it takes a higher temperature for a certain species to bleach and kick out their symbionts. And sometimes individuals within the same species have different levels of tolerance as well. So coral bleaching is kind of this environmental attack on the network. And when we typically model ecological networks and look at the stability of the ecosystems, we remove nodes as a simulation of species extinction. So we're going through and we're perturbing the system by taking out individual species and seeing kind of how robust is that network to that kind of perturbation. However, coral bleaching is the breakdown of the interaction between the host and its symbionts. It's a link removal problem. We're not having the corals or the symbionts go extinct, they're just no longer interacting. So coral bleaching is an environmentally driven process that results in a physiological breakdown. So we have to have information about the environmental condition of the system, so mainly the temperatures that the coral is experiencing. And we have to have some sort of information about the physiology of the different coral species and their different symbiont species, because they have varying thermal tolerances. So I assigned different thermal tolerances to the nodes in the network. So the node thermal tolerances are just the fraction of the maximum temperature increase that a node can handle. And it ranges from 0 to 1. So a node with a tolerance of 1 can basically won't bleach until we've reached the maximum amount of temperature stress that a coral can handle. If it's down at 0, then that coral is just bleached as soon as it gets too hot on the reef. So my research studies the effect of bleaching in the network by incorporating environmental and physiological data, and then simulating link removals to determine the resistance and robustness of the system, where resistance is that system-specific measure of stability. Here, it's the network's resistance to temperature stress specifically. And robustness is the general ability of the system to withstand a disturbance in the form of removals. Now, if you're an ecological network scientist, this is your typical definition of robustness. So I hypothesized that thermal tolerances, network structure, and spatial scale of the network will affect the resistance of the network. I don't have time to cover the spatial scale aspect of this research today, so I'm only going to be talking about the global network. Though in my paper, I do go through all 14 subregions and their resistances. And then I hypothesize that the different removal types, whether you're removing nodes or links, susceptible or resistant nodes or links, and generalists or specialists removing by high or low degree, will affect the robustness of the network. Now, I determine resistance and robustness of the system by developing a weighted link removal model that simulates bleaching on the network, where the weight is just a measure of the strength of the interaction and serves as a threshold for the breakdown of that interaction. And it's specific for each host and symbiont pair. So in the model, as temperature increases, if the weight of a host symbiont pair is less than that temperature, then it's removed. And once a host node is isolated, it's considered bleached. If it's isolated, that means that it no longer has any symbionts, so it's bleached. So this weight equation is based on 3 facts from coral biology. Now, the first is that corals start bleaching when temperatures are above the mean monthly maximum sea surface temperature. So basically, it has to be hotter than what they're used to for them to bleach. So the first term in the weight equation is the baseline temperature set by the sub-regional location of the host. Now, this is why it was important to include multiple nodes for species that were found in different locations, because it affected their weight equation. The second bit of coral biology that's informing this equation is that there is an upper thermal limit for the symbiosis, and this is set to 3 degrees Celsius. So the second term in this equation is the amount of temperature stress needed to break the link. And then finally, there's still a significant debate over who is most responsible for the failure of the symbiosis, the host or the symbiont. So the tolerances are averaged. So this weight equation reads as the baseline temperature plus the temperature stress needed to break the link, which is affected by the tolerances of both the host and the symbiont. Okay, so to determine how the structure and the thermal tolerances of the global coral symbiont network affect the network's resistance to temperature stress, I created null networks that change the original network in specific ways. So these null networks serve as a kind of control. The first 2 null networks change the thermal tolerance distribution. So the first null network, the shuffled tolerances, changed the tolerances of the nodes to new values within the thermal tolerance distribution. So it's asking, do specific thermal tolerances matter? The second one, the random tolerances null network, changed the tolerances to be picked from a random uniform distribution. So this one's asking, does the thermal tolerance distribution found in nature matter? The last 2 null networks change the structure. So the random bipartite degree conserved null network randomized the links, but kept the original network structure the same. So we still have that truncated power law degree distribution. So it's asking, do specific interaction patterns matter? Or if I go through and I shuffle the links, what happens? And then the final null network, the random bipartite not degree conserved model, changed the structure of the network to be random. So no longer that truncated power law. So does the network structure matter for resistance of the system? Okay, so I ran 100 simulations of the bleaching model on the global network and its nulls networks, and it spits out these response curves of the percent of hosts bleached as a function of temperature in degrees Celsius. To get a measure of resistance from these response curves, I defined resistance as the increase in temperature from 10% of hosts bleached to 90% of hosts bleached, normalized by that maximum temperature increase to 3 degrees Celsius. So it gives me a nice 0 to 1 value for resistance. Now, this metric could be adapted to a different system. It's just a measure of the amount of perturbation needed for the majority of the network to become disconnected when you're looking at the resistance to a specific stressor. All right, so here we have resistance on the vertical axis and the network and its nulls along the x. So this first bar is the resistance of the original global coral-symbiont network. When tolerances were shuffled within that same distribution, the resistance did not change. So specific tolerances do not matter. You can go through and change the tolerances around the network and the resistance does not change. However, when tolerances were changed to be from a random uniform distribution, the resistance increased. So yes, the tolerance distribution found in nature matters for network resistance. A random distribution made it more resistant. So the way it is right now makes the network, the system, more susceptible to increasing temperatures. Remember, these next two null networks changed the structure. And the resistance increased in both null networks. So the specific interaction patterns in the network structure do matter for network resistance to temperature increase. So having that truncated power law degree distribution decreased the resistance of the coral symbiosis system. A quick note on the stats, I used randomization tests to determine the statistical significance between the model results. I can answer questions about that later. So these were the results for the resistance. Now getting into the robustness of the network. So what is the robustness of the network to a removal? Here I use the common measure of robustness, the R50 value, which is just the proportion of nodes or links removed that results in a loss of 50% of the total number of nodes in the network. So I ran several different link and node removals. I removed links randomly or according to the bleaching model or by susceptibility, where link susceptibility was either the average tolerance of both the host and the symbiont, or just the host tolerance, or just the symbiont tolerance. And then I also removed nodes randomly, or by how many associations they have, or by their tolerance value. All right. So here are the results of the removal models on the global network, with percent remaining as a function of percent removed. But instead of looking at these curves, let's just jump right into the R50s. The first obvious difference here is among R50 values is between the link and node removals. The network is more robust to link removals than to node removals. Within the node removals, we see what we expect when removing by degree. When you attack the hubs, the network falls apart pretty quickly, but it takes a lot of removals of the specialist species to make a dent in the percent remaining. Also interesting within the node removals, the network is less robust when susceptible nodes are removed than when nodes are removed randomly. And then getting into the links, the first major result is that the network is less robust to removals according to the bleaching model than to random link removals. So there's something about this targeted bleaching removal model that makes the network less robust than if you were just to randomly remove links. And then also the network's resistance is lower when susceptible hosts are removed than when susceptible symbionts are removed. So this might indicate that it's the symbionts that are giving a bit more strength to the interaction than the hosts are. Okay. So To wrap everything back together and kind of go through the results for this project, the global coral symbiont network is susceptible to climate change. We can map the global associations of corals and their symbiotic algae as a network. And this network has a heterogeneous structure, like the airport network, the internet, plant and pollinator networks, which means that it's vulnerable to specific attacks, including bleaching modeled as a link removal process. Another vulnerability of the global coral symbiont network is its distribution of thermal tolerances. And within this thermal vulnerability, it's really the susceptible hosts that decrease the robustness of the network. All right, so the way that the coral-algal symbiosis network is in nature right now makes it decrease its resistance and robustness compared to null networks and the other types of removal models. So my research shows that the structure of the associations and thermal tolerances of coral species and their symbiotic algae are important considerations for coral reef conservation under global climate change. But kind of the big takeaway here is that is the importance of modeling ecologically relevant perturbations on network, because I found that the interaction of an ecosystem gave it more stability than the individual components, unless those interactions were susceptible to a certain stressor, like in the case of coral bleaching. So the global coral symbiont network was susceptible to thermal stress because of its structure of associations and distribution of thermal tolerances. And this is pretty bad news for coral reefs, especially when coral bleaching is getting worse. The number and severity of coral bleaching events are increasing and becoming more widespread. But luckily, corals don't always die from bleaching. They can recover if temperatures come back down to normal levels. So if the waters cool back down, the corals will actually take back up symbionts, or if there was some background level of symbionts still hanging out inside the coral, then those symbionts will be allowed to reproduce and kind of recolonize the coral. And then just like different species and individuals of the same species have varying susceptibilities to temperature stress, they also have varying abilities to recover after bleaching. So by limiting our spatial scale and expanding our temporal scale, we can start to answer questions about resilience and answer the full network plus stress equation. So I studied a local reef scale coral symbiont network in Bocas del Toro, Panama over a year through 2 heat stress events to determine resilience of the coral-algal symbiosis. And within this project, I was also trying to determine potential differences in coral host symbiont communities across host, depth, and time, and then determine possible differences in structural and functional resilience of the reef system. So I further break down resilience in this project into 2 different types. Oops, let's go back one. All right, so the first is structural resilience. It's a system's ability to either resist changing structure of associations or at least return to the initial structure after a disturbance. So this would be the case if corals bleached and recovered to associate with the same symbiont species as before they bleached. The second type of resilience I consider is functional resilience, which is an ecological system's ability to recover from a disturbance in relation to its health and function, but it may also undergo some structural changes during that recovery. So corals can actually have a flexible symbiosis, so that during or after a bleaching event, their symbiont communities can shift to either new species or they can shift the relative abundances of the species they already associate with. So those links are changeable in the network. This research took place at the Smithsonian Tropical Research Institute in Bocas del Toro, Panama. And the reef I chose is called Mogul's Reef. It's located outside the inner bay. If you're a marine scientist, that's important because the corals in this area gets a bit more water flushing in from the larger Caribbean Sea. So they tend to be a bit happier and healthier. So the study took place from January 2017 to 2018 on Mogul's Reef. I set up 200-meter squared quadrats, each at a different depth. The first quadrat was a shallow quadrat at 20 to 25 feet, and then the second one was a deep quadrat at 35 to 40 feet. At these quadrats, I surveyed 8 species, 8 different Caribbean coral species, and I tagged 3 to 5 colonies of each species at each depth. So by tagging corals, I mean I use the cow tags that they stick in cows' ears to keep track of cows, you can also attach those to corals. So you can go back and find the same coral that you monitored last dive. So that's what I mean by tagged colonies. This reef experienced abnormally high temperatures from April to December in 2017. So here on this graph, it shows the sea surface temperature and in situ water temperatures for both depths in degrees Celsius, as well as the degree heating weeks. This is the red line, which is just a measure of how hot it's been for how long. So you can see there's these two peaks, and these are the two major bleaching events that the reef experienced. So I initially surveyed and sampled in January 2017, and then the reef experienced these two peaks, one in June and one in October. So I surveyed and sampled during each of those events. Lastly, I returned to survey after temperatures had cooled down to sample at a recovered time point. So how do you collect a coral network? At each time point, I would take samples from each of the tagged colonies using a steel hole punch or clippers. So I would just end up with a really tiny little fragment, about a 1 centimeter diameter fragment of each colony. And then these small pieces of tissue were then immediately put into DNA preservative on the boat. And I ended up with a lot of tubes of coral juice. And so these coral juice were sequenced to determine what Symbodiniaceae species each sample had. You could just ignore the bioinformatics stuff. If you have questions about it, I'm happy to answer at the end. So what happened on the reef during this really, really hot year? So this figure shows the fraction of tagged colonies for each species that were either apparently healthy or visibly bleached. Given the abnormally high temperatures on the reef during the survey period, tagged colonies of every species surveyed showed visible signs of thermal stress that ranged from pale to completely bleached. You can see in the pictures of the same colony here, this is it during heat event 1 where it's completely bleached. Heat event 2, it was still a bit bleached, but it actually recovered during that time point a bit. So corals at the shallow quadrat showed more frequent signs of bleaching than those at the deep quadrat. And this is pretty typical for coral ecology. Corals in shallower waters typically experience more variable and higher temperatures. So they typically show more bleaching signs. So we've found what we would expect. Surprisingly, I didn't find significant differences among species, and this might be because of the sample size. However, from my personal observations and other surveys I did in the area, both Agaricea species, Porites asteroides, and Siderastua siderea were among the most susceptible species. So there were some varying levels of susceptibility on the reef. My data just did not capture it. And even though we saw a ton of bleaching on this reef, there was luckily a lot of recovery. So there was significant recovery at both depths, which is pretty cool for the network analyses. But before we can get into the network analyses, we have to talk about the, kind of these standard community analyses that are done when you're looking at the system. So here we have the non-metric multidimensional scaling NMDS analyses based on the Bray-Curtis dissimilarity between Symbodiniaceae community structures across coral hosts within the two different depth quadrats. So each point on the plot is a sample, and that sample is colored by the species of the coral. The closer 2 points are on the plot, and the more similar their symbiotic communities are. And then the ellipses are the 95% confidence ellipses for each species in ordination space. So this NMDS plot shows strong differences among species at each depth, which is supported by a per-MANOVA analysis relating the Bray-Curtis dissimilarity and symbiont abundance of the samples. So host species identity had the strongest effect on symbiont community structure, and then depth followed closely after that. Time was also a significant factor, but it wasn't easy to parse out from traditional community alpha and beta statistics. So to determine the temporal variation in these community structures, I turned to network analysis to clarify those temporal patterns. So I created symbiotic co-occurrence networks using the R package Speakeasy. And Speakeasy uses the sparse inverse covariance estimation for ecological association inference method for network construction. So it creates correlation networks, but it reduces the spurious results that are produced by traditional correlation analyses. So in the Speakeasy constructed network, a link between any two nodes implies that the taxa abundances of that symbiont species are not conditionally independent, and that there is a relationship between them that cannot be better explained by an alternate network construction. So this should be a pretty good network kind of construction for the symbiont communities at that time point, because I created networks for each time point and each depth. So these networks are just the symbiont species, and there's about 77 unique symbiont species present in the networks. All right, so scrolling along the bottom, I have GIFs for both the deep and shallow quadrats. So it should be scrolling through the different time points. So in the temporal symbiont co-occurrence networks, there were 71 to 77 nodes in all networks and about 112 to 165 links per network. And all networks had a fairly low connectance, the fraction of all possible links realized in the network. And then all networks were significantly more clustered than random networks. However, clustering also decreased through time and more for the shallow quadrat. So this clustering, the biological kind of thought of clustering would be that symbionts became less likely to co-occur with the same symbionts as their neighbors over time. So clustering actually decreased by half at the shallow quadrat and only by 1/3 at the deep quadrat. Those networks were becoming kind of more dissimilar. But to really get at the dissimilarity of the networks through time, I used a graph distance metric, the Hamming graph distance. And this graph distance measures the number of link substitutions needed to make one network topologically the same as the other. And it's just that measure of network dissimilarity. So it compares the overall structure of the networks created at the different depths at each time point. So looking at the correlation plot here, the shallow networks are more similar to each other than they are to the deep networks and vice versa. And then the deep networks were more similar across time than shallow networks. And of the shallow networks, the recovered time point network was more dissimilar from the other time points. So this is given by the color heat code. And that really the recovered time point network was most dissimilar from the heat event 1 network. So the structure of the network of symbiont co-occurrences became more dissimilar through time, and more so at the shallow network, where really the deep network was a bit more structurally resilient. It didn't budge as much. Okay, so from this project, I learned that host identity and depth drive the major differences among symbiont communities and samples, which varied through time as well. And the network analyses were able to clarify the temporal patterns in the data. So I show that a reef in Bocas del Toro that has experienced multiple bleaching events in the last decade, as well as during our survey period, may not have increased tolerance to thermal stress. We saw a ton of bleaching, but it was resilient. The majority of our surveyed corals recovered from bleaching. So that's that functional resilience. They recovered from bleaching. And then at the deep quadrat, the network stayed structurally sound as well. Next, we could move from the reef level to a species level idea of resilience for this project. So here, I know the font is pretty small for these, and I haven't gone through the analyses yet, but we can theoretically look at the individual species networks through time. So we would have the individual colonies of one species that we're looking at, and then the symbionts that species is interacting with, to then figure out, okay, so individuals of the species that bleached, what makes them different than the individuals that didn't bleach? And using network analyses to clarify those patterns. Additionally, I sequenced a subset of the samples for 16S to determine the bacterial communities of the samples. So this will give us a better picture of the entire coral holobiont, where the coral holobiont is just a fancy word for the whole organism. So it considers the coral host, the algal symbionts, and the bacteria. So in the microbiomes of the corals, I again saw strong species differences as shown by this NMDS plot. And then we can look at the co-occurrence networks of the algal symbionts and the bacteria to see how adding in the bacteria impacts the structure of the networks and the holobiont, and our idea of the resilience of the holobiont during these two heat stress events. All right, so getting back to our network plus stress equals question mark framework to kind of expand our view from coral reefs to other ecosystems. The way I set up my research and the way these two projects were really set up as a framework, we have our network, and it's really important to characterize our network in a biologically and ecologically relevant way. So we want to use physiological properties to inform the network characteristics. And we can do this by developing new methods to understand the physiology of a system, the individual components of the system, measuring stress susceptibility. So there's two really great meta-analyses that informed the thermal tolerances of the first projects, corals and symbionts. And then also looking at the temporal and spatial variability of a system helps inform the ecosystem, the network's properties. And then when we add in stress, we have to consider ecologically and biologically relevant stressors. Whether we do this by informing manipulative experiments that help us understand networks responding to stress, or carefully planning environmental monitoring into ecosystem studies, or developing new modeling frameworks like the weighted link removal model that I use to model coral bleaching. And then on the right-hand side of the equation, we have to look at both the resistance and the resilience, because resistance is kind of halfway to resilience, right? It's what's happening before the system starts to break down. And the resilience is whether or not that system's going to recover or adjust its ability and its activity to be able to recover. So an interdisciplinary approach that can combine knowledge of both physiology and ecosystem dynamics on local and global scales is needed to predict ecosystem responses to especially in environmental changes due to climate change. And this is really important for coral reefs as they are very highly threatened by climate change right now. All right, so that's all I have for you guys. And thank you for having me. I'd like to thank my funding sources real quick and all of my fieldwork help. And I'll take any questions.
You get a clap from me. Thank you. This was super cool. So I have a ton of questions, but I'll gladly let other people ask theirs. I'll just get started with something that I might have missed in the beginning. So we're talking about resilience and resistance and robustness, and all has to do something with a loss of functionality in one way or another. So what was your measure of functionality for your studies? Was that kind of overall connectance, or how did you measure it?
It kind of varies by study. So generally in ecological network science, we think of kind of the connectance of a network as kind of the holy grail for that network's functioning. That system's, you know, everything connected and working in the ways that it should. So when you're studying the robustness of a system, you're removing parts and seeing how quickly that network falls apart. And that's been our typical understanding of the robustness of these systems. And that's where I took robustness to the level of resistance to look at that system specific measure instead. And the resilience is looking at one step further, is if we allow the system to recover, how do we measure that? Yeah.
Right.
Right. And then with corals, we split that resilience into the structural and functional, where usually in ecology, we have the Holling 1973 definition of resilience, which is where if a system doesn't change its structure, you know, that's being resilient. So that's where I break it into structural resilience, and then functional resilience, the ability to adapt and change, deal with that stressor, and then still persist on a functional level later.
So can you see that, like if things change, say over the course of a heat event, and then you have a different network structure after the event, whether that network structure does its job just as well as the old network structure did?
I would need another year and a lot more funding.
You would need
I think if I went back and resampled those same corals, or went back just to monitor them to see if they're still alive, or measured other physiological properties before and after, like their photochemical efficiency or respiration rates, like doing other physiological measures to determine, like, is that coral still performing as well as it used to based on its new network structure? And I don't have those answers yet, but that would be really cool to look at.
I would think so too. I can't make the funding work out, but I can invite you back in a year's time. So that
Okay.
we can. I would also have more questions. I'm just trying to make sure that I'm not stealing away the shop for anybody else to ask questions over here. There's one in the chat. Would you like to read it out yourself? Can you access the chat?
Yeah, I can read it. So, thank you. So the question is, does removing one host, cutting itself off from a symbiont, result in overloading other hosts? Can that be considered as a secondary stressor? So not in the case of the coral symbiosis. It might be more appropriate for like a parasitic ecosystem, where if you took out a host, maybe that parasite went over to the other host and became more detrimental to those hosts. But in the coral symbiosis, removing a host doesn't really overload the other host with more symbionts. They don't just move over to another host. They're really with their own host. So they're not going to switch over. Yeah. So it's not really a secondary stressor in that way. But really interesting question for maybe a different kind of bipartite system. Yeah.
Is there some way that one could consider something acting as a secondary stressor in the coral reef networks?
If you threw in another environmental stressor like ocean acidification, people do large tank experiments where they kind of control the temperature and the pH of the conditions and look at how the coral's physiology is changing, and they normally look at also which symbionts are there and when or whether or not they lose their symbionts. And it's kind of gone back and forth, but it seems like ocean acidification actually promotes the symbiosis a bit longer under heat. Yeah.
Interesting. So it might be the opposite of a secondary stressor then. So if
Right.
my neighbors suffer, then I get to live longer or do my thing a little bit longer.
And then also maybe even if you expand our network from just the coral and its algal symbiont to its coral-algal symbiont and bacteria, how the environment is interacting, how the microbiome of the coral is responding to the environmental changes, that's kind of that secondary stressor as well. Because the algal symbiont and the bacteria are kind of inhabiting the same area of the coral. So they're kind of competing for space and nutrients and everything else.
Okay.
So a lot of interesting research being done with what happens when corals are bleached, what's going on with their microbiome. So that's part of the reasons I'm really excited to get into the microbiome data. I'm just not a microbiologist, so I don't know as much about the bacteria yet. But getting into that data will be pretty interesting to see how the entire holobiont is responding to that environmental change. And maybe the bacteria and what they're doing could be a secondary stressor. Because a lot of times after a coral bleaching event, that microbiome is thrown out of whack and you see coral disease happen because a beneficial bacteria that is normally really good for the coral under normal conditions now becomes pathogenic, and it will cause problems for the coral host. So that would be something where you have coral bleaching, which is the breakdown of the interaction between the coral and the symbiont, that then later down the line ends up being a problem with the coral and its bacteria. Yeah.
So
like a cascading effect. But yeah.
It sounds like a very, very complex system, very much so. I'm again stealing everybody's time, but I keep, oh, there's one more question by MC Ramos.
So what kinds of features make a coral species a hub? I have not looked into that. There was 2 papers out by Fabina et al. That looked at the geosymbio database from a network perspective in a slightly different way. And then they were looking at, why certain species were hubs. But I did not do that since they already had. And I think they were specifically asking questions about how coral species acquire their symbionts. So different species can either get their symbionts from their parents, or they can actually take up symbionts from their environment when they're still like a little baby coral that hasn't settled yet. So I think their hypothesis was that the corals that got their symbionts from the water column when they were a baby, instead of getting them from their parents, might be more of the hub species. Yeah.
Hi, I have a question that almost actually follows pretty well from
Okay.
that one. So I think as always, your research is really cool, and thank you for talking about it. I think your presentation was super well organized, and it made it really easy to understand exactly what are your nodes and what are your links.
Yeah.
I wanted to see if you could expand on the nodes and links and how the relative populations of different corals might affect this, just because relative populations of things is what I'm kind of used to seeing in terms of change over time of ecological systems. And here you have these networks instead. So yeah.
Right. Yeah. So that would be a different way of formulating the networks, of constructing the networks. The way I've kind of done it is this more, I'm going to use meta network approach, but someone's going to ding me on that. It's fine. So here I'm just thinking about what's like a species' ability to associate with symbionts. This is what we know, like known associations. So we have the coral species and the symbiont species, and whether or not we see any sort of co-occurrence of those two. The way you're talking about it, we could look at an entire patch of reef, right? And that's kind of what we're doing, what I did in the Panama project, was looking at 100-meter squared area of reef and looking at those individual colonies and seeing what they associate with. So you could go through and do a more population-based study like that, where you say, well, this is my sample, let me expand it to a larger reef scale and model it kind of in that population dynamic way, where then you could have the meta population where each patch is different, or it's really like scaling down your networks. And that's definitely possible. It's not something I've explored yet, but would be really cool to do, kind of moving into these different levels of coral networks, whether you're looking at one individual colony and its associations or possible associations, and then moving up to a small patch of reef, and all of the different coral colonies would be different nodes and different symbionts. But then you can group that into one node really to see what is that population doing. That's what usually ecological kind of studies do in that kind of meta population framework, and counting that as one node in the larger node of more areas, if that makes sense, sort of.
Yeah, definitely. Yeah. And also yeah, it's an interesting situation, because you don't really get a lot of like big swings in population, do you? Especially not if symbionts are something that a colony or a polyp gets right at its start and doesn't change, then it would be.
Yeah. Well, they can change during and after bleaching. But the problem is that the host isn't going anywhere, because the host is so long living, hopefully, right? Like it can die now because of us. But the idea is, normally corals are very long-lived creatures. They're not going anywhere. That they're not really mixing with other populations. So their symbiont communities should be pretty static under normal conditions, right? And so the host is static, and the symbionts do change around a bit. Yeah.
Mm-hmm. Awesome. Blowing my mind over here.
Mine too. So I kind of one thing that I definitely wanted to ask you before we break off is, first of all, with coral reefs under stress, is there something that we can learn from your research about how we can save the reefs, other than that we really should do that? Like, is there, for example, applications in bioengineering reefs to become more resilient in the future? And secondly, what's next for you?
What's next for me?
Yeah.
Well, first, there's this whole area of coral biology looking at growing supercorals. And the idea is that you are kind of bioengineering certain pairings of individual corals based on their genetics and their ability to withstand stress, along with making sure that they're paired with like the strongest symbionts. But then I also show in my overall model that there is a bit of a danger doing that, because you have to know how the whole system is doing, not just one species and how that one species is surviving, right? So you have to kind of scale that up to a larger scale, really. So moving up scales, as always.
Is there a danger by putting supercorals out into the wild that they steal the kind of habitat of non-supercorals, or are they not that much in competition?
I think it would depend on where they're going and how dead the reef is where they're going. The problem is that there's really just not that much live, healthy reef anymore. So it probably doesn't hurt to put them out on the reef. But if those supercorals are susceptible to a stressor that we might not have predicted, like maybe they're super susceptible to a certain pathogen that comes across that reef, then you're wiping out that entire reef because you just had all these clones out there. So it's something you have to watch for. Yeah. So there is this way of maybe doing kind of a network thinking of coral restoration, and just kind of including that in the way we plan our management efforts. And then, so I guess what's next for me, this is only a small, well, it's a medium subset of my research on corals. Otherwise, I look at the networks of individual coral polyps and how they're connected. And then right now in my postdoc, I'm really focusing on disease models for coral reefs. So there's the stony coral tissue loss disease outbreak in the Florida Keys and in the wider Caribbean. And my work's really focused on modeling the disease outbreak in the area right now. Yeah.
That is super cool. So I am looking forward to seeing where your research takes you, and for updates on you and your puppy. And thanks again for talking today. This was absolutely awesome. And thanks to everyone who joined. And I'll see hopefully most of you again next week.
Yes, thank you so much, everyone. Thanks for being a great audience, and thank you for having me.